Hi everyone, my name's Tom Rollins and I'm the marketing manager here at FORMULATRIX. I'd like to take the opportunity to thank everyone very much for taking the time out of your busy schedules today. I'm extremely excited for this application-focused live web event. Before we begin, I'd like to provide an overview of the agenda. First, Paul Burning, our North American sales manager here at FORMULATRIX, will briefly introduce solutions for positive displacement microfluidic reagent dispensing for RNAi screening workflows. Then, Chi Yoon, director of the High Throughput Biology Lab at NYU Langone Health, will explain the importance of transfection optimization for high-quality RNAi experiments. Chi couldn't be with us today due to an unforeseen circumstance, so we recorded her talk late last week, and you'll be hearing it for the first time here. Next, Dan Bradford, laboratory manager from the Stowers Institute of Biomedical Research, will explain his research and how the Mantis has enabled automated and miniaturized Gibson assembly and PCR in his lab to promote efficient and reproducible RNAi screens. At any time in the presentation, please feel free to enter your questions into the GoToWebinar interface, and we will answer all that we can at the end of the live web event. With that, I'll now introduce Paul Burning. Paul is our North American sales manager here at FORMULATRIX. Go ahead and take it away, Paul.
All right, thanks, Tom. So first of all, I too would like to thank everyone for attending our webinar today to hear about RNA workflows in a biological lab. As Tom mentioned, I'm Paul, our North America sales manager. And during my presentation here today, I'd like to tell you a little bit about FORMULATRIX, our products, and our role within labs all over the world. A part of what we do here at FORMULATRIX is educate. So as I go through these slides, I'd like them to be educational in manner and not just come across as a quote unquote sales pitch. But I do like to point out the value proposition. All right, so here we go. So when I talk about FORMULATRIX, I always like to start with our why. And this first sentence could not be more true, and I could not say it any better myself. I know it's against presentation etiquette to just read the sentences on the slide, but I do see this as an exception, so bear with me here. FORMULATRIX enables scientific discovery by designing solutions without boundaries and bringing novel cutting-edge technology to the life science industry. We design innovative products that create their own market and add value for scientists. So what I mean by that is we bring engineers to the scientists to drive science forward. As you can see in our employee chart here, almost half of our employees are engineers. And that is true with almost 500 employees at FORMULATRIX. I did want to touch on our product offering from FORMULATRIX. We have two pillars to the company. If you go on our website, you will find that there is a protein crystallography pillar and then the liquid handling and digital PCR pillar. I represent the liquid handling and PCR side, as you can see by the products listed here. To date, we have these three products on the market, but we do hope to launch 3 or 4 more by the end of 2019. The first is the Mantis liquid handler, which Chi and Dan will be discussing in their workflows of RNAi. The Mantis is a non-contact, low volume, low dead volume reagent dispenser. The Mantis has the capability of dispensing droplets down to 100 nanoliters. We use a positive displacement technology, which I will explain on the next slide. But since it's positive displacement, we are able to achieve 1.4% CVs at these low 100 nanoliter volumes. That reproducibility at low volumes allows scientists to miniaturize their genomics applications, whether it's PCR or NGS. And as you can see in the photo, the Mantis has the ability to use a pipette tip as a reservoir to achieve a dead volume as low as 6 microliters. The big brother to the Mantis is the Tempus. The Mantis dispenses into a single well at a time, whereas the Tempus will dispense into 96 wells at a time. To put this into perspective, the Mantis will take about 25 seconds to fill an entire 96 well plate with 1 microliter, whereas the Tempus will perform that same dispense in about 3 seconds. So it's extremely faster. The Tempus uses the same positive displacement technology as the Mantis, which once again, I'll go into detail on the next slide of how the diaphragm technology actually works. One of the main differences, though, between the Mantis and the Tempus is the lowest volume dispense. The Mantis will go down to 100 nanoliters, while the Tempus will go down to 200 nanoliters. As you can see, the plate stackers in the photo, the Tempus is usually geared towards higher throughput settings or fully automated systems, although both instruments are automation friendly. All right, and last but certainly not least, we have the Constellation. The Constellation was just launched this past January and is a fixed partitioned digital PCR platform. It is an award-winning platform. During this past SLAS, we were one of 3 winners out of 15 submissions to win the new product award. It is something that we are very proud of here at FORMULATRIX. I won't go into the technology too much because I'll do that on a later slide, but the idea is that we're going to offer absolute quantification of your targets as opposed to using reference curves and assumptions in conventional PCR. With this technology, we plan to disrupt the legacy PCR market by being more cost-effective and offering higher quality data at a lower cost. We feel that if we can win in these 3 areas, speed, data, and cost, the decision becomes a no-brainer to adopt the technology. All right, and I'd like to go through how our diaphragm technology actually works. In the heart of our technology are our microfluidic chips. This is a high volume chip designated by the green back here. Inside each of our chips, we have 2 diaphragms or microfluidic valves. The smaller valve in the high volume chip is 1 microliter, and the smaller diaphragm on the high volume chip is 5 microliters. On either side of the diaphragms, we have an input valve and an output valve. And the Mantis actually uses a sequence of positive and negative pressure to actuate the input-output valves and the diaphragms. So positive pressure is designated by the red here, and then negative pressure or vacuum will be designated by the green. Once again, we use the sequence to drive your solution through the chip. The middle diaphragm is going to be the volume that you're looking to dispense. So once again, in this particular chip, it's the 1 microliter, 5 microliters. So we fill this same diaphragm over and over again, which is how we're able to reproduce those droplets. From there, we close the input valve as the solution comes through, apply positive pressure, open the output valve, and then the droplet comes through. The low volume chips, the 2 diaphragms are going to be 100 nanoliters and then 500 nanoliters. So the resolution of the technology is going to be in 0.1 microliters. And you can find that video on our website as well. All right. As I mentioned on the previous slides, the target applications for our products lie within the genomics market. The Mantis will handle many areas within NGS library prep, including tagmentation and amplification steps, along with normalizing your library that you're going to pool them together. Since the Mantis and Tempus use positive displacement, viscosity plays a low role in the precision of the dispensing. Therefore, master mix dispensing in your PCR workflow fits right into the wheelhouse of our technology. The Tempus is a premier product when it comes to dispensing cells. We offer recirculatory feature that keeps your cells in suspension to ensure even dispensing into your plate. Our software is user-friendly. If you are looking into assay development or DOE, you are able to input in concentrations, and our software will calculate the volume that is needed to be dispensed. The main goal of any application when it comes to our technology is to offer a solution to process your samples with confidence and within budget. Of course, we are going to hear Chi and Dan speak about how they use the technology, so I won't go into the RNAi workflow, but the Mantis and Tempus will certainly fit into the editing and screening portions, while the Constellation has the ability to confirm those edits. All right, speaking of the Constellation, I like to go over this technology before I hand it over. I like to describe the Constellation as 2 different technologies, 1 being the instrument itself and 2 being the plate technology. So I'll start with the instrument first. The instrument is the first fully automated plate-based digital PCR instrument to hit the market. You can simply load your qPCR reaction into our plates, put a mat on the plate, and load the plate onto the instrument, and then walk away. The instrument then primes, thermocycles, and images the plate all without human intervention. We have different plate formats for different applications, depending on the amount of sensitivity you may need. One of the benefits of having different consumables to fit different needs means that we can easily engineer different plates to fit changes in the market. We do not have to redesign the instrument, and we do not have a one-size-fits-all. We currently have 96 sample by 8,000 partition plates and look to release the 24 sample by 36,000 partition plate after a software update before the end of 2018. The instrument is considered scalable to fit whatever your throughput need is. You can build the system to have 1 or 2 trays to hold 4 or 8 plates, as well as 1 or 2 thermocyclers. The instrument is also considered high throughput. The software will schedule the priming, thermocycling, and imaging to achieve the fastest results as possible. If your thermocycling conditions are 60 minutes, we are able to process the first plate in around 90 minutes, and then an additional plate will come out every 15 minutes following. We can process all 8 plates in under 5 hours. So load up all 8 plates and truly walk away. FORMULATRIX doesn't sell reagents, therefore you can use your current vendor. The master mix we use in-house when customers send in samples is NAD, but we have used every other commercial master mix on the market. And lastly, we have the ability to multiplex your samples and get results for up to 5 targets. We currently use rocks to determine which partitions are filled, therefore it's going to be our reference dye. All right, now I'd like to explain the plate technology for everyone. This is the entire plate. We're going to zoom in on one well. The image on the left is a bird's eye view of the plate, and the image on the right is going to be a horizontal view. The input well is this circle on the bottom left of the bird's eye view, and it's going to be this chimney. You pipette in your reaction. So if you're multiplexing 5 targets, you're going to have all your probe primer designs. You put your mat on the plate, and then you load the plate onto the instrument. The instrument then primes it, which means a piston comes over, presses down on the mat, and the reaction floods the partitions. A roller then comes across the bottom and separates the partitions. So now we have 8,000 little PCR reactions, if you will. From there, we thermocycle, where the reaction will take place. And then from there, we image and count the positives and negatives. Using the Poisson statistics, we then determine the copies per microliters, which can be converted into any concentration value if you know the length of your amplicon. All 96 are done in parallel. So this is what 96 samples would look like for one filter. If you are multiplexing with all 5 filters, you'll get 5 times this amount of data off of a single plate. All right, looking at the applications that the Constellation fits in. For the 8,000 partition plate, we have customers looking into CNV, gene expression, and gene editing confirmation, as well as sequencing validation, SNP detection, and library quantification for pooling. Once again, the 24-well plates will be released shortly, but those plates will be used for rare target applications for those researchers that are looking for the needle in the haystack. All right, thanks, Tom.
Thanks so much, Paul. That was a really nice overview of the technology and how it can benefit researchers performing those target applications. Next, we have a presentation from Chi Yoon from the NYU Langone Health Center. As director of the High Throughput Biology Laboratory, formerly the RNAi Core, at NYU Medical Center, Chi has overseen hundreds of diverse 2D and 3D cell-based high throughput screening projects from academic researchers in the New York City area. As an expert in functional genomics, high-content screening, and cell-based assay development, she has presented at high-content analysis meetings, directed courses on best practices for RNAi screening, and teaches a graduate course section on automation, screening, and data analysis. She co-founded the NYU Image Analysis Working Group, which discusses software applications, image analysis standards, project-based examples, and best practices for data retention. She is also chair of the Mid-Atlantic chapter of the Laboratory Research and Innovation Group, a special interest group focused on laboratory automation and new technologies.
Hello, my name is Chi Yun, and I am the director of the High Throughput Biology Laboratory at NYU School of Medicine. And today I'll be presenting my thoughts on transfection optimization for successful RNAi screening. As a shared resource lab open to both academic and for-profit institutions, we offer expertise and services for a variety of cellular and biochemical assays. The core was first established as an RNAi screening core, and as such contains specialized equipment to support projects ranging from custom to whole genome screening. We have liquid handling instrumentation that range from simple plate fillers to programmable discrete dispensing with the Mantis, to large-scale robotics integrated with barcode printing, labeling, and heat sealing. Our assay instrumentation comes in two flavors, plate readers and imagers. We have infrared, monochromator, and filter-based readers, as well as a Seahorse XFE analyzer for real-time live metabolic measurements. For imaging, we have high-throughput automated imagers that offer concurrent analysis with image acquisition. Several in-house libraries are available for screening. These include an FDA-approved compound library and whole genome and custom siRNA microRNA libraries for human, fly, and mouse. Our tissue culture is equipped with both standard and controlled O2 incubators, and hoods contain equipment for sterile cell dispensing and media exchanges. Finally, the High Throughput Biology laboratory staff offer flexible project support ranging from equipment training and consultations to full-service large-scale projects. So I'd like to emphasize that while our name is High Throughput Biology, in reality, we welcome projects ranging from a single plate to hundreds, one data point to complex multiparametric analyses, new assays to tedious low throughput experiments. Absorbance, fluorescence, and luminescence are plate reader technologies used in many of our RNAi screens. Our multi-mode plate readers have been used for high throughput ELISAs, cell health and proliferation, and other cell-based assays in both compound and RNAi screening in both 2D and 3D cultures. Other RNAi screens performed in our lab utilize our high-throughput, high-content capabilities. Some assays are fairly straightforward, such as colony counts and measurements or target analysis. Others are more complex, such as multiplex toxicology assays to characterize mechanism of action or involve 3D co-culture models. The type of RNAi screening that we perform in the HTV lab is arrayed library screening. In contrast to pooled library screening shown here on the left, where deconvolution of the reagents is required at the end of the screen, on the right, in arrayed screening, the identity of the reagent within each well of the assay plate is already known. Our RNAi libraries are pre-plated with the siRNAs in 384 well plates, and we perform reverse transfection, where diluted transfection reagents are first complexed with the siRNAs, followed by addition of the cells. Here I've outlined the general workflow of RNAi screening. First, you begin with an optimized cell-based assay, and with the RNAi library, perform the high-throughput primary screen. This is followed by data analysis to identify primary screening positives, followed by validation and secondary screening to end up with a finalized hit list. For a whole genome screen, you're typically starting off with testing the knockdown phenotypes of about 23,000 genes, which is equivalent to 210 384 well plates if screening in triplicate, with the goal of narrowing down your primary screening positives to about 100 to 300 genes. Many users are often in a rush to begin screening. However, this very first step, optimizing the cell-based assay, in particular the transfection optimization, is absolutely critical to the success of the RNAi screen. One basic consideration when developing your assay is the choice of cell type. The cells should reflect your biology of interest, and transfection should be optimized to the desired phenotype, which may or may not strictly correlate with maximal knockdown. The assay choice should also be relevant to the biological question, but might also be constrained by other factors. For example, loosely adherent cells might not be ideal for antibody staining in an image-based assay. The use of both positive and negative controls are essential, with a non-targeting control indicating background, and ideally a positive control as something specific to the biology. We have had successful screens where a general control was used to assess knockdown, for example, GFP siRNA with cells stably expressing GFP. However, assay-specific positive controls at varying degrees of strength allow for easier thresholding of positives during analysis. Lastly, in particular for plate readers assays, for reporter cell line, cell normalization is critical. Now, the goal when optimizing your cell-based assay is to define reproducible conditions where you can have a quantifiable, robust phenotype. As I mentioned previously, efficient transfection is critical. Cell line passage and overall health are very important in finding the best transfection conditions. Since there can be some toxicity caused by the transfection process, you want your cells healthy and happy to measure the downstream biology. Things to consider include mycoplasma contamination, passage number, confluency at the time of cell preparation, and possibly including immediate change 12 to 24 hours post-transfection. Also, we generally find that reverse transfection works as well, if not better, than forward transfection. Lastly, testing cell number and different reagent types and amounts are critical for transfection optimization. Some users strictly rely on vendor-provided or lab-established protocols. However, the same cell line can have different sensitivities to the reagent, or the amount used might not be best for the assay endpoint. Because of this reluctance to test so many conditions manually, we have incorporated the Mantis for the transfection optimization process. In our general reverse transfection protocol, we dispense 5 microliters of diluted transfection reagent into a 384-well plate that already contains 5 microliters of a 0.3 micromolar siRNA pool. We spin down the plate for complex formation and then add 40 microliters of cells at the appropriate dilution. Using the Mantis, we can quickly test multiple conditions in a single plate. In the following example, we test 1,500 cells versus 2,000 cells per well, 5 different amounts of 4 different transfection reagents, and also use 3 different siRNA as controls. The scrambled siRNA acts as a negative control, the KIP11 siRNA causes a cell death phenotype, and the incept a multinucleation phenotype. So shown here is the plate layout of the siRNAs listed in each well. Each filled color represents a different type of transfection reagent. We make diluted master mixes consisting of 1 microliter of reagent plus 4.9 microliters of Optimum per well for each of the reagents. By adding 5 microliters of this mix to a single well, there is a 0.1 microliter final amount of the transfection reagent. In order to test decreasing amounts of this reagent, we backfill with Optimum to keep the total volume of the siRNA and diluted transfection mix constant. Shown here is the final map with each shaded cell color representing a different reagent and the number listing the final amount of transfection reagent used. Plotted here are the KIP11 and scrambled siRNAs versus valid object count or cell number, trellised by the reagent type and amount. You can see that at 1,500 cells per well, for all 4 reagents, the difference between scrambled and KIP11 is greatest at the lowest amount of reagent, 0.01 microliters per well. You can also see, for all 4 reagents, increasing toxicity, the scrambled siRNA with increasing reagent amount. The other control siRNA used in this experiment was INSEP, whose knockdown should result in multinucleation. Plotted here are the INSEP and scrambled siRNAs versus mean nuclear area, trellised by the reagent type and amount. Overall, the difference between INSEP and the scrambled siRNAs is not as dramatic as KIT-induced cell death. For reagents 3 and 4, multinucleation does not track as closely with the cell death phenotype, but this could simply be due to the timing of the assay endpoint. So in conclusion, transfection optimization is a critical component of assay development, and automation speeds up optimization and allows testing for multiple parameters. Taking the time to optimize your screening assay from transfection to assay endpoint is really essential for a successful RNAi screen. A garbage assay will result in garbage data. If available, assay-specific controls are best. General controls might not be the best for transfection optimization, as you can see based on the discrepancies between the KIF11 and INSEP siRNA data for reagents 3 and 4. Finally, keep in mind that you want to maximize your assay robustness and reproducibility so that you can easily find hits in your favorite pathway or biology. Thank you for watching, and feel free to email me at chi.yun@nyulango.org with any questions or comments.
Great. Well, I'd like to thank Chi very much for the very informative talk on transfection optimization. Please note that we will forward any questions over to Chi that come through, and we'll get to you promptly with answers. So please feel free to type any questions that you have into the GoToWebinar interface. Now, I'm excited to introduce Dan Bradford. Dan is trained as a molecular biologist, started out developing genotyping assays for paddlefish and validating microarray data. He has spent the last 13 years working with laboratory automation systems to develop high-throughput genetic screens. Take it away, Dan.
Okay. Hi, everyone. I'm Dan. I'd like to thank FORMULATRIX for giving me an opportunity to present our work here. I'm going to talk about a high-throughput screen we've developed with automated liquid handlers to develop the knockdown of genes in the process of tissue. Getting a little bit of slides. There we go. I manage an at the Stowers Institute for Medical Research in Missouri. Our institute was founded in 1994 and began research in the year 2000. Now, Stowers is a research institute. Our founder, Jim, also founded American and left a significant part of his fortune as our endowment. We have about 500 institute members in 21 research labs spread across core labs and administrative positions. The focus and strength of our institute is the collaborations between core facilities and research labs. When you come here and do your work, you don't have to be an expert in every single discipline. As a result of this, papers published here often have long authorships. So within the molecular biology core facility here, we have 3 teams. Each has a different focus. The next-gen and Sanger sequencing team, the genome editing team, and the automation and PCR technology team, which I manage. Our mission is to support the 21 research labs here through provided services, collaborative projects, as well as independent research and development of emerging technologies. I've been working in automation here since 2006 and took over management of this group in 2012. We collaborate with research labs here to develop and complete high-throughput automated projects with a variety of lab robots. We have 3 TCANs, 1 biomech, a QPix colony picker, a yeast gritting robot, and a variety of high-throughput analysis instruments that enable us to successfully complete these projects and screens. Over the years, our group has contributed to 15 publications, of which the most prominent are these 2 papers in Nature and a third in Cell. We also support qPCR, which utilizes an additional 2 liquid handling robots, as well as droplet digital PCR, which incorporates liquid handling, microfluidics droplet generation, a thermal cycler, and a flow cytometer that reads the droplets. To help describe how the RNAi pathway works, I'm going to do a very brief review of molecular biology's central dogma, in which DNA is transcribed into RNA and subsequently translated into protein. That's it. End of review. At Stowers, we perform basic research, which is to say that our focus is to determine what genes and their protein products do. One way to do that is to knock out or knock down gene function and observe the effects on phenotype. RNA interference uses an already existing cellular pathway to knock down expression of specific genes. When double-stranded RNA enters a cell, a surveillance protein complex called dicer, which isn't shown here, detects the presence of double-stranded RNA and cleaves it into small molecules. At that point, the RISC protein complex binds the small single-stranded RNA molecules and presents them in a way that, due to the complementary base pairing properties of nucleic acids, can only bind to specific RNAs with a complementary sequence and targets them for destruction. As a result, the messenger RNAs are destroyed, which inhibits the translation of the protein product. And the model organism that we are using to investigate RNA interference is the planarian Schmidtia mediterranea. Our particular colony came from a water fountain in Spain. It was actually lost once in the past prior to our collaborators moved to Stowers due to filtration issues, and the lab had to return to the source to reestablish it. However, this fountain has since been removed, so ours may be the last living colony from there. Planaria are an ideal model organism to study tissue regeneration due to the abundant pluripotent stem cells called neoblasts present within them. As a result, these organisms can be dissected in a variety of ways, and they can regenerate back into a complete organism within a matter of days. If we can fully understand and harness this regenerative process, this could have very profound implications on regenerative medicine. So I'd like to give a little background here so you can appreciate the evolution of this type of screen. Early screens simply injected double-stranded RNA directly into the tissue. This produced a localized and a temporal effect. Our collaborators then moved on to plasmid expression vectors, but had to abandon gateway technology simply due to its high cost. They then developed ligation-independent cloning, which entailed doing PCR on cDNA, where the 5' regions would have restriction enzyme sites. This required cleaning up the PCR reactions to remove the primer dimers and unincorporated DNTPs, a restriction digest, a T4 polymerase reaction to add sticky ends, and then these inserts were added along with the vectors into the transformation reaction, and you would hope that once they got inside the E. coli that its own repair mechanisms could produce a fully intact RNAi expression vector. Modern screens now use the soft serve technique, in which these transformed E. coli are mixed with beef liver and then fed to the planaria. The worms are almost all gastrointestinal cavity, so the RNAi vector is delivered very uniformly throughout the organism, which is shown here by the worms having ingested beef liver along with blue and green food coloring, respectively. Now, the drawbacks to this method are it's really labor intensive, and something that's labor intensive isn't necessarily easy to automate with robots. It's also got a high reagent cost due to the cost of restriction enzymes and T4 polymerase, and our efficiency of transformation just was never very high. So that meant lots of picking and screening. So we'd like to screen the entire genome of these planarians, which is 20,000 genes. To do that, we needed to make some improvements. The first thing we did was move to a Gibson assembly strategy to generate these recombinant plasmids. With this strategy, there's no need for restriction digest or T4 polymerase reactions. And we found that if we did 2 rounds of PCR on the cDNA and overloaded the vector with the insert, that our transformation efficiency just went through the roof. We also moved to a magnetic bead PCR cleanup after the second round of PCR that was very robot friendly. Gibson assembly was then performed with clean PCR inserts and a linearized vector produced by PCR. All of this was done in a single tube in a single reaction. The Gibson assembly reaction was then added to component cells in a 96-well plate and transformed before plating onto these 48-well segmented bioassay trays. And as you can see here, our efficiency is pretty good. We have quite a few colonies in each one of these sections to pick from. And now our colony picking robot would go into action and pick colonies, which are then screened by Sanger sequencing. Once we had this high throughput pipeline in place, we realized that we had a need for a low volume dispenser. The large liquid handling instruments are great at the jobs that they're designed to do, but there are things that they simply struggle with. Among these are dispensing small volumes into dry wells. And this is no trivial task, as anyone who's had experience with automated liquid handlers could tell you. Repetitive aspirate and dispense steps can also be a challenge, especially when you're talking about 96 or 384 sequential steps. And tip-based dispensers require dead volumes in reservoirs that is basically just wasted reagents. So as stated previously, we're doing 2 rounds of PCR in 96 well plates. The Mantis can dispense from 50 mil conicals, as shown here, and that eliminates the need for tips. We're currently dispensing 23 microliters per well, but we're looking at decreasing this to a 10 microliter reaction in 384 well plates. We've been able to reduce the manufacturer's recommendation of a 20 microliter Gibson assembly reaction down to 5 microliters. The Mantis enables us to dispense 3 microliters of Gibson assembly master mix into dry wells of a PCR plate. And tip-based dispensers would struggle at this. And I took a picture here. I hope that you can see this is actually 3 microliters that is dispensed right into these PCR plates, and they're all uniform. Costs for our 96 well tip boxes for our different liquid handlers vary from $2.50 to $5 a piece. And so the savings to this project over 20,000 genes would be substantial. And just some basic math here, if I throw it together, we're saving anywhere from $2,500 to $5,000 just on this single project. So this instrument would pay for itself pretty fast. So I'll conclude with a couple of interesting hits from this technique that we've been able to replicate in our high throughput RNAi cloning pipeline. When beta-catenin is knocked down and the worm is dissected, it grows back 2 heads. When APC is knocked down and the worm is dissected, it grows back 2 tails. Now, the largest published screen, RNAi screen in Planaria, is around 1,000 genes. We're at 6,000 genes currently, and we have about a 3% hit rate. And I'm not going to share any novel hits that we've had until we've concluded the screen and published, but that hit rate is pretty solid. We have some pretty good leads here. And so just to wrap up, I'd like to acknowledge Alejandro Sanchez. He's the PI whose lab that we're collaborating with. Frederick Mann is his postdoc who's driving this work with us. Some past people that we've worked with, Beth Duncan, who now has her own lab at the University of Kentucky, Aaron and Stephanie, who are still at Stowers, and the person that I started on the screen with the ligation independent cloning, Carrie Adler, who's now at Cornell with her own lab. Andrew, he's the research technician in our lab who's really taken the Mantis by the horn and is using that to drive more productivity with the screen. And Karen, my director here within the molecular biology core facility at Stowers. Thank you, and we'll take any questions.
Thanks so much, Dan. That was an awesome presentation. I'm really excited to see how the Mantis has enabled a significant cost savings in your lab. So if you just give me 1 second here. Okay, so we have a question. It looks like for you, Dan, is the Mantis integrated into any of your larger liquid handling platforms?
We don't have it integrated right now. We do have some plans to acquire a new liquid handling robot, possibly mid-year next year, and we are thinking about integrating it into that system.
Very interesting. And I have another question here about liquid classes. I think that could either be for you, Dan, or Paul. Do you have to program liquid classes for the Mantis?
They have.
I can go ahead and take this. Or go ahead.
Yeah,
Go ahead, Dan.
No, you're fine. You're probably more qualified to answer than me.
So with the positive displacement, we recommend, or we market that we're able to handle viscosities up to 50% glycerol with a plug-and-play manner. So there should be no need to calibrate it, whether you have a reagent up to that equivalent. But if you're above that, what we will do is we can slow down the timing of the sequences and then increase the positive and negative pressures to make sure that we're filling the diaphragms completely to make sure that when we do actually dispense the droplet, that is a full droplet. So short answer, no liquid classes, but if you do have a very high viscous solution, we can optimize it.
Great. So there's another question here about dispensing cells. Does the diaphragm technology shear cells?
Yeah, it's another great question, actually. So in my experience dispensing cells, we have not seen any shearing with our base settings. But once again, if you have a cell that may be fragile, we can change those parameters to make sure that the impact of the diaphragm on the solution, whether it's your media or water, is low. So then as the cells are moving through the diaphragm technology, it's not going to have a high impact. Or when the droplet actually is placed into the plate, it's at a lower speed as well. So, I don't want to say no, right? But I will definitely say that in my experience when working with customers, it does not cause shearing.
Okay. So I have a logistical question here about the presentation slides and whether I can send them out. So this webinar has been recorded and we will send out the recording following the event for everyone. So one question here, how long does it take for the Mantis to dispense into a 96 well plate?
So is that for me?
Yeah, go ahead if you know the answer.
So for our PCR reactions where we're dispensing 23 microliters, it takes about 40 to 60 seconds to dispense across the 96 well plate. But for the smaller dispenses, our 3 microliter reactions, it takes probably 10 to 20 seconds.
Okay, so that's great. Thanks, Dan. I have another question for you here. Have you lost any sensitivity when you reduce the volume of your PCR reactions?
No, we haven't. But then we're using we're not just loading these on a gel, so it might be more difficult to detect on gel type technologies. We're using LabChip GX and the Advanced Analytical Fragment Analyzer, so they're pretty sensitive.
Great. Okay. Well, that's all of the questions that we have time for today. So I'd like to thank everyone again so much for taking the time out of your busy schedules. I hope this was an educational event and that everyone take something away from this. And I'll be sure to send out the recording soon. Thanks again, Dan, and thanks, Paul, for your time and for putting together a couple of really nice educational presentations.
All right. Thanks, Tom. Thanks, Dan.
Thank you.